So let's move to the next poster. This work is entitled Nanotechnology, a tool for a sustainable insecticide delivery. The work is done by Catarina Ganilho from the Green Porto institution from Portugal. So Catarina, the floor is yours.
So I'm sharing my presentation. Can you see it?
Yeah, sure.
So good afternoon to everyone. My name is Catarina Ganilho. I'm a master's student and I'm working at Green Porto Research Center. And I'm glad to be here to presenting the topic nanotechnology, a tool for a sustainable insecticide delivery. So population growth has to lead an increase and demand for food, and consequently the intensification of agricultural activity using insecticides for the protection of the crops. But despite the benefits and the need for agri-food sector in pest control, the excessive and widespread use of insecticides can cause environmental contamination. For this reason, several environmentally sustainable strategies have been proposed for the control of agricultural pests. One of the strategies is the application of nanotechnology that has benefited the sector by providing the use of nanopesticides, in which lipid-based nanomaterials allow the encapsulation of insecticides and a controlled release of active ingredient of these insecticides. However, given then their complexity, ecotoxicological studies of lipid nanomaterials are often controversial, and thus detailed investigation of the effects on the environment is required. So for this reason, this work aimed at evaluating the effects of the active ingredient of insecticide lambda C halothrin encapsulated in lipid nanoparticles on different non-target organisms. And this work as well allows the safety to be assessed for non-target species, and then to select the doses that are not harmful to non-target species to be tested for efficacy in target organisms. Thus a safety by design perspective is applied. So we tested our nanoparticles on different aquatic organisms like Daphnia magna, microalgae Rhaphidocelis subcapitata, and aquatic plant Lemnos minor. And you tested on terrestrial organisms like Colambula species Folsomia candida. For aquatic organisms, the assays were carried out with elutriates prepared from samples, sediment samples containing lipid nanoparticles loaded with lambda C halothrin, while for the test with Colambula Folsomia candida, the soil was directly homogenized with lipid nanoparticles loaded with lambda C halothrin. And the concentrations tested were based on the doses of lambda C halothrin applied in agricultural practices. So the methodology, the lipid nanoparticles were synthesized based on physiologic lipids and without the use of organic solvents. And the synthesis follow a method of a hot homogenization using an emulsion oil water as a template. First, we add the aqueous phase to oil phase, both at the same temperature, around 75 degrees. The lipidic, the oil phase, contains the lambda C halothrin because the insecticide is lipophilic. After this, we homogenize strongly for 10 minutes to create an emulsion oil water. And the last step was cooling the emulsion in an ice bath to create the lipid nanoparticles. As you can see here in this figure, the lipid nanoparticles loaded with lambda C halothrin. And we characterize our nanoparticles concerning the particle size. For this, we measure the particle size at the moment of the synthesis, the day 0, and 1, 2, 3, and 4 days after the synthesis. And our results shows that the mean particle size of our nanoparticles was around 165.4 nanometers, and the particle size was stable for 4 months when stored at the room temperature, around 25 degrees. Regarding ecotoxicity studies and first the acute immobilization test with Daphnia magna, The methodology, so the test was assessed using 5 Daphnia magna neonates under the age of 24 hours, as you can see here in a figure 2 with our experimental test design. The Daphnia magna neonates were exposed for 48 hours, and the neonates were as well closely watched during 24 and 48 hours. And at the end of the assay, the percentage of immobilization was calculated. And the data obtained showed that the percentage of immobilization of Daphnia magna neonates was 50% after 48 hours of exposure at 23 lambda cialotrin grams per hectare. Now the grow inhibition test with the microalgae Rhaphidocelis subcapitata, the methodology, so the microalgae were exposed for 72 hours. And at the end of the assay, the growth inhibition rate was calculated. And with our results, we can conclude that the microalgae growth was stimulated in all concentrations tested of lipid nanoparticles low with lambda cialotrin when compared to the control group. The grow inhibition test with aquatic plant lamina minor, the methodology, the lamina were exposed for 7 days. At the end of the assay, the number of fronds represented on the graph with the color dark green and their dry biomass represented on the graph with the color light green were recorded. And at the end of the assay, the growth rate was calculated. And the results demonstrate that the growth of lamina minor was not affected by the presence of lipid nanoparticles loaded with lambda cialotrin. The last study, the reproduction test with Columella falsamia candida. The assay was performed in plastic containers, as you can see here in a figure 8 in the first image. In each container were added 10 falsamia candida juveniles, and they were exposed for 28 days. At the end of the assay, we take a picture of the vessels, as you can see here in a figure C, and you take a picture to count the number of juveniles using computer software. And the results show that the reproduction of Columella falsamia candida was not affected by the presence of lipid nanoparticles loaded with lambda cialotrin. So now our conclusions. So the data of the present work allow concluding that this project allowed us to follow the safety by design perspective to assess the safety of our nanoformulations and the concentrations that could be safe for non-target species to be tested for efficacy in target species. And the results showed that the lipid nanoparticles that we synthesized can be an excellent strategy for the encapsulation of insecticide lambda cialotrin because the particle size were stable for 4 months, the reproduction of Columella falsamia candida and the growth rate of aquatic plant lamina minor were not affected by the presence of lipid nanoparticles loaded with lambda cialotrin. The percentage of immobilization of Daphnia magna neonates was 50% after 48 hours of exposure at 23 lambda cialotrin grams per hectare, but this concentration value is higher than an actual concentration applied in the field. And the microalgae growth was stimulated in all concentrations of lipid nanoparticles tested. However, further ecotoxicity studies are being carried out with other soil organisms, for example, reproduction tests with other soil invertebrate Ysenia fetida, growth and germination tests with the plant of Venasativum solemn Lycopersicum, and also we are testing microbial community to evaluate the effects of these lipid nanoparticles. So now my acknowledgements. Thank you for your attention. I finished my presentation.
Thank you very much, Catarina, for the presentation. Let's open the questions and comments from the audience. If there's nobody, I have a question. It was surprising nanotechnology that in my research field of engineering, it's another kind probably of nanotechnology, but it's nice to see that it's not only in engineering that we are talking that engineering, my electrical engineering. So it's nice to see that. So you are observing that it's not affecting in a bad way the use of these nanoparticles of pesticides. So, but have you studied what you mentioned that during 4 months it's not affected the size of these nanoparticles? So
Yes.
it means that it maintains the same conditions. But have you studied more than 4 months? Is affected?
Yes, because the nanoformulation becomes jelly, so we can't measure the particle size. So
okay, so that's like a caducity date for that kind of pesticides. So it's like for any other content. Okay. Right, so and now we are trying with more other plants and other soil.
yes, other
animals.
organisms.
Okay, nice. It would be interesting to see the results after this test that you are performing.
Thank you.
Any other comments, questions to Catarina?
There is one comment in chat.
Let me check. Sorry. Manon says that they don't have any questions, but thank you for this presentation. It's a discovery for me. So.
Okay, thank you for your interest and the opportunity to present.
Okay, so thank you again. Thanks for joining us in this workshop and congratulations for your work.
Thank you.